All right, so today we're going to finish up biochemistry. On Wednesday, we talked about carbohydrates, lipids, and we finished up talking about amino acids. And we said that they're essentially the building blocks for making proteins, and that there are 20 common ones. And we said in the body, the carboxylic acid end typically is deprotonated, so that oxygen has a negative charge, and the amine is protonated because it's a base, so it has a positive charge. And we call this as Witter because it has both charges on one molecule. I don't want you guys to memorize these, but I did want you to know how to interpret these tables. So in general, these amino acids have a R side chain, and on this table they're highlighted in green. And we tend to clump amino acids into groupings based on whether or not they're hydrophobic, hydrophilic, or basic or acidic. So let's talk about making proteins instead of just individual amino acids. So when we make a protein, all we're going to do is take two amino acids and try to link them together. So really this is an offshoot of polymerization that we've talked about already. So I'm just going to draw two generic amino acids. All right, so I've got one of them over here. And I've got an R group. And I've got my carboxylic acid on the right-hand side. And over here, I've got my other amino acid with another generic R group and a carboxylic acid. If we're doing a condensation polymerization, what small molecule do you think we're going to lose? Water, which is a good thing because it's happening inside our body. We don't want to produce hydrochloric acid or anything weird like that. So really, these two pieces are going to link together and you're going to shoot off a water molecule. So I'm just going to show a dipeptide. I'm not going to continue on and show a full continuing chain. Okay, so essentially this is the new bond that we've made. And this bond has a very particular name. Does anybody know what this is called? It's a peptide bond. So polypeptides, you can think of a peptide bond linking together amino acids. All right, let's do a little bit of practice though. What's this new functional group called that we've made? We've got a carbonyl with a nitrogen on one side. Not quite. An ester would be an oxygen attached instead of a nitrogen. Amide. Yeah, exactly. So we made a new amide functional group by linking two amino acids together. And you can keep on doing this over and over again and get these super big, long polymers of polypeptides. And that's essentially the building blocks of our proteins. So let's talk about how they link together. And I've got this drawing here. You guys obviously don't have to copy this down. But you can see in the structure we've linked together a bunch of amino acids. So what I want to do is identify all of the peptide bonds. And peptide bonds are pretty easy to find because it's in between that CO double bond and your nitrogen, right? So they show them right here. We've got a peptide bond here, 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 and here. And then off of each of the individual monomers, or your amino acids, you've got these R groups kind of dangling off. And the identity of the amino acid is defined by this R group side chain. So for example, let's just look at this first one. We've got an R group that's a CH3. Sorry, I can zoom in. It's a little hard to see. Okay, so a CH3 side group. So let's go back up to our table and see if we can find out what amino acid that would be. So I'll scoot around. Oh, if we look at alanine, it's got a CH3 R group, so that means it's an alanine base unit in that peptide sequence. Does that make sense? This is referred to as the primary structure of polypeptides. It's basically just the order of amino acids as they are linked together. So let's make a note of that. So primary structure is the order of amino acids. However, we know our proteins aren't just big long chains. They actually adopt unique shapes and orientations, and so we have to describe these secondary, tertiary, and quaternary structures as well. So let's jump into secondary structures. A secondary structure is a little bit different than your primary structure. Essentially, it describes how that primary structure folds into a unique shape. So polypeptides adopt unique shapes along portions of their chain. And this is due to H bonding. So how do we define an H bond? Does anybody remember the easiest way to spot an H bond? Yeah, hydrogen that's covalently bound to fluorine, oxygen, or nitrogen. We don't see fluorine really at all in biomolecules, but we do see oxygen and nitrogen, right? So let's go back up and look at the structure. If we look at it, right, we've got an NH right there, we've got an NH right here. These are really, really sticky subunits. They want to stick to other protein molecules, and because of this, they adopt really unique shapes. How many of you guys have taken basic biology classes? A few people? How many people remember the unique shapes? There was one that started with alpha. What was that one? Alpha helix, right? So an alpha helix is one of them, where basically it just spins around into a helical staircase type structure. And then there's another one called beta sheets, where essentially you have two strands that will hydrogen bond together. And so you've got two unique portions that are sticking together. And then you'll have other portions that are just simply other forms of coils or coiling. And this is all due to hydrogen bonding, but it's basically describing the secondary structure of how proteins come together. So let's take a look at how you might see proteins drawn in a textbook. So for example, what do you think this big yellow portion is right there? Let's zoom in. You think that's a beta sheet or a helix? Helix, exactly. So this would be an alpha helix. Let's see if we can identify this portion right here. So can you guys kind of see that green one? What might that be? Beta sheet. So you can see it's not curving around as much. It's more just two strands together. So these would be beta sheets. So they adopt these really unique structures simply due to the hydrogen bonding. All right, there's also tertiary structures as well. So let's describe that really quick. I'm going to zoom out. The tertiary structure is basically looking at higher level folding. So basically a tertiary structure is the overall shape of a polypeptide strand. So for example, the tertiary structure could contain both alpha helices, beta sheets, things that aren't H-bonded, some coiling. It's basically just the overall polypeptide and how it folds. Quaternary structures are even more complicated. And a quaternary structure is basically multiple polypeptides forming a complex. So instead of just one polypeptide strand adopting a shape, it's multiple of those kind of glomming together into one big glob. So for example, in the structure up above, you can see we've got a blue polypeptide, a yellow polypeptide, a red one, and a green one. And they're all kind of docking together in order to form something more complicated. So that's our quaternary structure. Does that make sense? So proteins are really important in biology. They're our building blocks, but they also do their own unique reactions. Does anybody know the name of a protein that catalyzes a reaction? What do you think? An enzyme, right? So I'm sure a lot of you have heard about enzymes, right? So coenzymes or enzymes. So these are really unique proteins. An enzyme is a protein that catalyzes a chemical reaction. So, second question, what the heck does catalyze mean? Does anybody know? If you think about the speed of a reaction, if you add a catalyst, does it go faster or slower? Faster. So it speeds up a chemical reaction. And it doesn't get consumed. So essentially a catalyst, like a catalytic converter in your car, is converting potentially harmful gases into relatively benign gases, but it regenerates itself every time it does that. So the catalyst is, in theory, recyclable, right? So these enzymes are pretty unique. Typically an enzyme is thought of as a lock and key system, right? So an enzyme might have a little pocket on the inside that's got a very specific shape. And that specific shape is designed so that only one thing can fit inside of it. And the thing that basically docks with the enzyme is referred to as a substrate. So you can see up here we've got this substrate floating around, and this enzyme's got a pocket that's roughly the same size as that substrate. So the substrate will essentially dock inside of the enzyme, and we call that our enzyme substrate complex, is when they're both kind of docked together, just like when you put your key in your door. Only your key fits inside your door lock, hopefully. After that point, the enzyme will do a reaction. So over here you can see maybe this enzyme is going to cut this piece right here, so it's going to cleave that part off, and then basically spit it back out into products. But our enzyme at this final step is back to normal. That means that once it's back to normal, another equivalent of substrate could come along, dock in the center, and a new reaction can occur. So these are basically really intricate biomolecules that are designed for a handful of specific reactions. If you go on and take further biology classes, you'll talk more and more about enzymes, but they're hugely important. Does that make sense? Okay, now we get to talk about nucleic acids. Nucleic acids are pretty neat. And we talked about this briefly the other day. What are the two types of nucleic acids in our body? RNA and DNA, right? So let's do DNA first. DNA is the most common one you hear people talk about. And DNA is alphabet soup for deoxyribonucleic acid. So if you look at it, it makes sense that we call it DNA because this is a mouthful, so DNA. And then our other main nucleic acid is RNA, where it's just ribonucleic acid. So how do you think they differ from one another, just by the name? What do you think? Yeah, RNA is missing an oxygen at some point. And so we'll talk about that. It's really easy to spot RNA versus DNA because RNA has an oxygen in a spot where DNA doesn't. The easiest way to describe DNA or RNA to people is that this is our genetic hard drive. So just like your computer has a hard drive to store information, we have genetic information stored in DNA and RNA. And it basically has the blueprints for your body. They do some other stuff as well. So for example, RNA can also do catalytic reactions, but for the most part, your nucleic acids are just there to store information inside of your body, right? Does anybody know what they code for? So RNA or DNA gets translated into what after?
mRNA.
Yeah, so DNA might go to mRNA, we'll talk about that, and then what does that RNA get translated into afterwards? Yeah, tRNA, which turns it back into a protein. So this is all interconnected, right? So basically DNA and RNA is just a code for what proteins go in what spots, right? Or what amino acids, I should say, go in what spots. So let's take a look at a simple nucleotide. And these are essentially our monomers. So DNA is made up of a bunch of nucleotides all linked together. And they've got three main components in them. They've got this phosphate head. And this is essentially going to be one of our main components of the backbone of DNA and RNA. And then over here, what biomolecule does that look like? Is it a fat? Is it an amino acid? Is it a carbohydrate? Looks a lot like our sugars that we talked about earlier, right? So this is our sugar. And in this case, this sugar is called ribose. And then over here, we've got this other cyclic structure with a bunch of nitrogens in it. This is called our nucleic acid base. Or I'll just call it a base. But the neat thing with the base is that it's really designed well to hydrogen bond to another strand. So how many of you guys have seen the structure of DNA? What structure does it adopt? Double helix. So you've got two strands sticking together. The bases are designed to hydrogen bond to another strand, which is kind of neat. We'll talk about that. So let's talk about our bases a little bit. Whoop. I don't expect you guys to remember the structure of these, obviously. But I do want you to know how they pair, and I'll show you guys some tricks for identifying that. So for example, in the structure I showed you guys, we've got this one. There's two rings in it. I'm going to zoom in a little bit too. So one of the rings is basically a 5-membered ring. The other ring on the right-hand side is a 6-membered ring. What base pair do you think this might be looking at this table? It's a little confusing, and you may have to spin it around. Adenine, right? If you look at these other two, they've got the CO double bond in one of the positions. The one I circled doesn't. So this would be an adenine base pair. And each base pair is designed to basically link up with another base pair, and we'll talk about how to identify those. So really, in DNA, you've got a certain set of base pairs. In DNA, you have thymine, adenine, guanine, and cytosine, right? So those are our main ones. So I'm going to make a list over here. And people get really tired of writing these out. So thymine, they just write with a T. Adenine, they write with an A. Guanine, they write with a G. And cytosine, they write with a C. All right, and then RNA is a little bit different. RNA doesn't have the exact same base pairs as DNA. So I'm going to zoom in a little bit. RNA will still have adenine. It will still have guanine. It will still have cytosine. But instead of thymine, you end up with a new base called uracil. So RNA and DNA are really similar, but they have one base pair that's a little bit different than the other. So I'm going to go through and make a new list for RNA. So instead of thymine, we said we had uracil. That was our change, right? other than that, basically everything's the same, where we have adenine, guanine, and cytosine. So that's the main difference in the base pairing difference between RNA and DNA. The other thing we said based on the name was that DNA, the deoxyribonucleic acid, is missing an oxygen. So if we look at DNA, the sugar in that monomer has no oxygen here, so no OH in this position for DNA, right? This is a deoxyribose. Where if we look at RNA, right? RNA does have an OH coming off of that lower right-hand position, so this would be the sugar for RNA, a simple ribose. So RNA has an OH here. Does that make sense? It's a little bit confusing. I do want you guys to know the base pair differences between these. So for example, I could say, all right, is thymine a base pair for RNA or DNA? And I would expect you guys to be able to say, oh yeah, that one's only in DNA, not in RNA. So that's really the core concept I want you guys to know from this portion. All right, so just like amino acids, we're going to polymerize these monomers together into one big long chain. So I'll show you guys how that works. So nucleotides polymerize into long chains. You can tell these get really, really complicated really fast, but the backbone of the polymer is basically sugars and phosphates. And then coming off of the backbone, you've got these other base pairs. And we said the base pairs could be thymine, adenine, guanine, or cytosine. It depends if you've got RNA or DNA. We said RNA has uracil instead of thymine, so it does vary a little bit, but the overall structure is really similar for both RNA and DNA. So let's talk about how these will link together. So we already mentioned base pairs like to hydrogen bond together in pairs. So base pairs can H bond together. All right, so if we look at this one over on the left-hand side, you can see we've got a hydrogen bond here and here. So two hydrogen bonds. This occurs anytime you have a thymine that is adjacent to an adenine, right? So I call this an AT base pair or a TA base pair. It can go either way, right? If we look at the ones on the right-hand side, we've got one hydrogen bond here, got another hydrogen bond here, and we've got another hydrogen bond here. So we've actually got more hydrogen bonding between these base pairs. And this always occurs between cytosine and guanine. So this would be CG. So these are referred to a little bit differently. So if you go on in biology, you'll talk about purines and pyrimidines, but adenine and guanine are typically thought of as purines, and the easiest way to remember these is that these are bicyclic. They have 2 rings. And these are different than the pyrimidines. Oh, sorry, this is kind of sloppy. So pyrimidines only have 1 ring. Oh. So they do vary a little bit. I'm not going to be super picky, but I did want you guys to know that A's and T's bind together, and then C's and G's bind together. All right, now we've got a kind of tricky one. All right, in RNA, though, we don't have any thymine, right? So with RNA, if you are forming a base pair, what would it be instead of T? U. So in RNA, it would be an AU pair instead of an AT pair. That's really the big difference between these. So let's take a look at the 3D structure of DNA. And I'll show you guys a little bit of history that really grinds my gears. So we already mentioned that DNA forms this double helix. And I'm going to put this in quotes. Who discovered the structure of DNA? Does anybody know these 2 people's names?
One of them is helix.
Not helix. That would be too perfect. It's Watson and Crick. So Watson and Crick were the first chemists that published a paper describing the overall structure of DNA, and they were basically proclaimed as the greatest scientists around, and everybody thought they were awesome. Does anybody know who really discovered?
It was this lady, I don't remember the name.
Rosalind Franklin. So Rosalind Franklin was the female scientist that did all of the experimental work to figure out the structure. She handed off all of the data to Watson and Crick, and they're like, great, we solved it. And it's like, wait a minute. I solved it. I just gave my data to you, right? So Rosalind Franklin is kind of this unsung hero of biology. She really should have won the Nobel Prize. Unfortunately, she died at a very young age, and a lot of people think that she is really the one responsible for discovering the overall structure of DNA. This was a really, really hard thing to figure out at the time. Chemistry was still fairly undeveloped in the 1950s. We didn't have great ways of predicting molecules. One of my favorite stories is there's this chemist called Linus Pauling. Linus Pauling is one of the only people to have ever lived to win 2 Nobel Prizes. He won one in chemistry, and then he won the Nobel Peace Prize. Linus Pauling was basically the person that helped develop chemistry as its own field, right? He also published a paper claiming to discover the structure of DNA, but was entirely wrong. It was a big embarrassment. It's like this guy won 2 Nobel Prizes and was so far off base that it's kind of funny to look at today. So it's one of those things where if you goof up, even the smartest people on the planet goof up on occasion. So I just like talking about this because Rosalind Franklin is hugely important in the discovery of DNA and RNA. The other thing, too, just to get back on my political soapbox here, this guy's still alive and he's a huge racist. So I'm just going to put that in. So in the 1940s, obviously, if you've studied anything about like Nazi Germany or even in the United States, there's this idea that people with certain genetics were superior than other people, right? He was also one of those people that continues to advocate this idea of genetic superiority, and that's just, it really irritates me, and I don't like him. All right, moving on. Just to reiterate, the main differences between DNA and RNA is that DNA has a sugar that's missing an OH. DNA is usually found in your body as a double strand helix, and the base pairs are adenine, thymine, cytosine, and guanine, so CGAT. With RNA doesn't have that missing OH. In fact, it has it where it should be, so it's just a simple ribose sugar. RNA is usually found as a single-stranded polymer. It doesn't hydrogen bond and form that double helix as frequently as DNA. And then the big difference we said here is that there's no thymine, there's uracil. So other than that, they're really similar molecules to one another. Does that make sense? I know we're moving through this kind of quick, but we're just trying to get through the basics here. All right, so let's take a look at just the overarching theme behind all of this, and this is protein synthesis. And scientists call this the central dogma. The central dogma is basically we've got this genetic information stored in our body as DNA. That's our genetic hard drive. And then this DNA is converted to a special type of RNA. This is called mRNA. Does anybody know what the R or the M stands for? Messenger. So the DNA is stored deep inside of your cell in this region called the nucleus, right? And in order to get that information to the factory that produces proteins, you actually have to transfer it outside of the nucleus into the cytosol. So this process of converting DNA into this messenger molecule is called transcription. And you have a bunch of cellular machinery that will actually read along your DNA and copy it over into messenger RNA. So at that point, your messenger RNA will kind of travel outside of the nucleus through these miniature pores, and then it will find something called the ribosome, and then it will get converted into a protein. This second process where mRNA gets changed over or read into a protein is called translation. The way I always remember this, right, is if you think about translate, it's from one language to another, right? The language of nucleic acids is completely different than the language of proteins, right? So translating that genetic information into protein information is translation. So let's kind of look at this big picture here. So inside this nucleus, I'm going to highlight this with our red line here. We've got our DNA. And this DNA at this step is getting translated into this messenger RNA. And the messenger RNA is kind of neat. It will get kind of fed out of the nucleus and float out. And then once it floats out, you've got this really special type of RNA that's basically floating around looking for messenger RNA. Does anybody remember that special type of RNA? tRNA. And that's for the translation. So right here, what we have is a tRNA, and this is referred to as transfer RNA. It converts mRNA to proteins. It's pretty dang weird in my opinion. So if you think about it, in your cells you basically have these bumper cars just constantly running into one another. So you've got messenger RNA and tRNA constantly bumping into one another. They will only form proteins if they kind of click together. And how do you think they recognize each other in order to click? How do biomolecules stick to one another? Think about DNA. How did DNA stick together? Hydrogen bonding, right? So again, this is all due to hydrogen bonding. It's a bunch of bumper cars. If they have the correct matching hydrogen bonds, they'll stick together and then actually form a big, long protein sequence. It's amazing how chaotic our bodies are, but how efficient they are at making proteins, right? So let's take a look at kind of what's going on here. We'll do kind of a game. So DNA codes for mRNA. Whoop. mRNA pairs with tRNA. Whoop. Holy cow. And then tRNA pieces together polypeptides. All right, so I'm going to make a list here of DNA, RNA, actually mRNA, excuse me, and then the tRNA, and then I'm going to put base pair. And this all requires things to stick together in the right order. So DNA, let's say I've got a cytosine. What must the mRNA base pair be in order for it to stick together? Guanine, a G. All right, and then going back out to tRNA, we need to find another complementary pair. What should that be? C. It kind of goes back and forth, right? So they have to match each other during this process. Okay, let's do another one. Let's say we start out with a G on our DNA. So we've got guanine. What must our mRNA base pair be? C. And what must our tRNA base pair be? G. So it kind of flip-flops back and forth. All right, let's say I've got adenine on my DNA. What must our mRNA be? Why not T? Because it's RNA. Good catch. Normally my class doesn't get that. So normally if it's a DNA base pair, it would be a T. But because we're going from DNA to RNA, it must be a U, right? So let's put a little asterisk there. This occurs because it's simply RNA. All right, and let's back out again. Let's say, all right, we're going from mRNA to tRNA. What must the base pair be? A. Back to A. All right, let's say we start out with thymine in our DNA. What must our mRNA be? Would it be a U? It'd be an A, right? So the only thing we switch is a T to a U in RNAs. All right? And now that we've got an mRNA with an A, what must the tRNA be? A U, right? Because it can't be a T in the RNA. So we'll say U, and these are the big things you got to pay attention to, is that T's get flipped to U's in the RNA. T is only in DNA. Okay, so let's take a look at this. So tRNA has a 3 base pair sequence called a codon. So instead of just one base pair, they're always in triplicates of 3. And you have to find a matching mRNA sequence. It's amazing how this works, in my opinion. So let's take a look at one of these interactions. Or shoot, this should be called an anticodon. Sorry about that. Okay, so this molecule right here, kind of this whole lower portion, is our tRNA that's floating around. So this is tRNA, and you can see it's got an anticodon with 3 base pairs. And you can see this C has to match up with the G, U has to match up with an A, and an A has to match up with the U. So they're really selective with where they bind on the messenger RNA. There's a ton of these tRNAs floating around in a cell. And it's really important because this anticodon basically tells you what amino acid is dangling off the end. So, for example, that anticodon is selective for valine in particular. So each tRNA has a specific codon, or I keep on saying codon. each tRNA, oh my goodness, has an anticodon that is specific for an amino acid. And the amino acid we could see in this one was valine, right? Okay, so let's take a look at a table here. And I'll show you guys this table. And then we'll come back to this. So this table basically tells you what amino acid belongs where. However, this table does get a little bit confusing. This code, so for example, I'm going to box out this UUU. This is our mRNA sequence. It basically says that the tRNA is looking for that specific sequence, and then once it finds that specific sequence on the mRNA, it will hydrogen bond to it, and then that phenylalanine hanging off the end will be the amino acid for that section of messenger RNA. So it gets really complicated. It's this intricate ballet of tRNAs coming together trying to find their correct spot along an mRNA. So let's take a look at it. So we've got this mRNA floating around on a cell. tRNA is kind of bumping into it, trying to find their correct spot. And you can see maybe in this mRNA strand, we've got 3 U's in a row. That means that the tRNA must have 3 A's in order to hydrogen bond together. And then that amino acid will get linked to the RNA to the left of it, or to the polypeptide to the left of it. And then the next tRNA will fit in and then link it together. Next one, link together. It's almost like forming a big long braid of amino acids as it's going down the chain. All right, so let's try to do another game with this. Okay. So I'm going to give you guys a DNA sequence. And then what I want you guys to do is to figure out the mRNA sequence. And then I want you to figure out what amino acid would correspond to that mRNA. So I'm going to do AGA, and then next to that I'll have an ATA. All right, so let's try to find our mRNA first. So for AGA, what should it be? UCU, exactly. So A normally goes to T in DNA, but it has to go to U for RNA. G is always paired to a C, and A again is going to be a U. All right, so now we've got to go to our table to figure out what amino acid is coded for with UCU, right? All right, so we go up, and we'd say, all right, our first base pair is going to be a U, so we know it's got to be in that row. Our second base pair has got to be a U, so it's going to be in this row. And then our second one's a C. So it's UCU, so PHE, that's phenylalanine. So I'd say PHE. So on your sapling, you may have problems where you have to refer to a table. On an exam, for example, on the final exam, I would give you a table to refer to help figure out this problem-solving sequence. All right, let's do the next one. ATA. What would that be? UAU, right? Because T can get flipped back to an A. All right, so now we have to find a UAU in the table. All right, so first base pair, again, is a U. Second base pair is an A, so now we're in this column. And then we've got a U. So it looks to me like it's got to be right here. PYR. So that's our specific amino acid, and we can plug it in right here. Yep.
The last one you had was a UCU, but that doesn't go to the chart. It's not PHE.
Oh my goodness. Holy cow. UCU should be serine. Thank you. Thank you for doing that. I was going way too fast. I would have missed points on my exam. All right, so yeah, make sure you go through and double check that. The other interesting thing you might notice in this table is you've got a couple of these sequences that are termination ones. Essentially what that does is it tells you, hey, stop making the protein here. This is our cutoff point, eject it off, and that's going to be the polypeptide. So that way we don't form one big long infinite chain, we're forming discrete polypeptides. And the other interesting thing you might notice is, let's take leucine, for example, in green on this side. This can be coded by a wide variety of mRNA sequences. So it's not one specific mRNA sequence for one amino acid. It can actually be a handful of them. But our body's done a really good job of figuring out this system selectively. So that's essentially where I'm going to leave it today. If you are interested in watching some videos to kind of see what this looks like, I've got some cool videos on here. It's kind of fun to see how the sequence works out, but I'll keep these on the OneNote notebook. But it's kind of this ordered chaos. I don't know. I find it kind of interesting and beautiful to imagine our cells being as chaotic, but as refined as they are.